So good morning, good afternoon, and good evening to all of you, depending on where you are. Thank you for joining us today. My name is Neringa, and I will be your host for today's webinar entitled Best Practices for Genetic Characterization and QC in Cell Line Development. So let's move to the first slide. Yes, perfect. So as you're probably well aware of, CLD is often regarded as the bottleneck within the pharmaceutical supply chain, as shown on this image. And in fact, the entire process usually takes several months. And current CLD best practices still rely on a series of laborious, resource-intense, and time-consuming processes. And with the rise of biopharmaceutical candidates in the pipeline, there is an ever-increasing need to streamline cell line development process to mitigate risk and accelerate early phase testing. Unfortunately, what we also sometimes see happening with some companies is that when their drug development program is near the finish line, and basically just when they are about to get ready for the clinical trials, they unexpectedly pick up a genetic sequence variant in their original MCB, which then leads to months of pushback on filing their IND. So therefore, the rapid and successful commercialization of your products will require you and your team to think about risk mitigation strategies early on and balance time and the resource appropriately. And in order to facilitate early phase testing, minimize time to clinic, and keep costs down. So in this webinar, we will go over the different stages of a cell line development process. Their proper genetic QC steps could be implemented. And at each of these critical steps, we will go over some of the key challenges and considerations. We will also provide the best genetic QC practices, and we will define the role of our TLA-based QC. So let's start with the optimization and validation of editing methods. So to date, biomedical researchers have an access to a wide range of genome editing technologies, and these scientific advancements over the years has enabled the possibility to directly target and precisely modify genomic sequencings towards better understanding the contribution of genetics to disease, thereby providing new insight into new cure and treatment strategies. All of these tools, of course, come with their own set of pros and cons. And if we take the example of CRISPR-Cas9, the system has generated a lot of excitement because it is faster, cheaper. It is also more accurate and more efficient than other genome editing methods. However, as revealed in a Nature Biotechnology article, scientists at the Wellcome Center Institute reported significant on-target mutagenesis, such as large lesions and more complex genetic rearrangements at the targeted sites. So underscoring the need to implement proper control strategies to assess the consequences of your genetic engineering. So coming back to your CLD work, whether you're applying a random integration approach or adopting more targeted strategies, you will want to evaluate the outcome of your genetic manipulation to ensure that the event you're after has actually occurred as intended. And ideally, you will want to do this as early as possible during your development phase process to ensure the success and accuracy of the gene editing approach and the quality of the resulting products. So on this note, Horizon Discovery recently applied TLA-based solutions to assess the genetic outcomes of their multiplex gene editing strategy. There are 4 different loci that are targeted simultaneously towards the generation of improved CHO cell lines. And TLA was used as a robust validation approach for several of the clones generated to identify all the genetic outcomes from the gene editing events. If you're interested in learning more about this study, I invite you to check out our co-hosted webinar via Gen News or download our joint paper. So while random integration is the conventional method used in the CLD process, we do see a shift in our client projects towards semi-targeted and targeted approaches, especially for the CHO cell lines. And we show a few examples here by leading biotech and pharma companies. Their TLA-based solutions have been used to help them perform comprehensive genetic characterization and QC. And in fact, the focus of industrial CLD seems to have shifted towards host cell line stability, the use of targeted integration techniques, and the development of selection and screening systems. As shown on this slide, these are typically the questions our customers are concerned with and come to us when initiating a clone selection phase. Early clone screening is important in cell line development since this helps to certify that the selected clone does not harbor any undesired genetic variants. Because DNA mutations or incorrect DNA replication and mistranslation during protein synthesis can give rise to defective proteins. Such events can affect the safety or efficacy of a final therapeutic product. Commonly, MS approaches such as LC-MS are usually the first ones used to retrieve information on location, identity, as well as relative quantification of sequences variants at protein level. These techniques, however, are laborious and yield low throughput, especially when detection of low-level sequence variants is required. Therefore, as a result, NGS presents itself as an attractive and alternative candidate in terms of speed, cost, throughput, and sensitivity. Therefore, they are increasingly applied in biopharmaceutical research and development. In 2019, Novartis reported for the first time the application and benefits of our TLA NGS-based workflow as part of their CHO clone selection and described how the method can process up to 24 CHO clones in less than 7 workdays. So as a result, this publication illustrates how comprehensive genomic analysis can greatly benefit speed and quality of medicine development programs. So again, selecting the CHO clone with a preferred genetic makeup is a crucial step in the cell line development process towards producing stable and high quality biotherapeutic proteins. And the necessity for early clone screening also continues to be heavily suggested by big biopharmaceutical companies such as Boehringer Ingelheim. If we move more downstream of a cell line development process, we reach master cell bank stage. A well-characterized cell bank ensures the consistent supply of equivalent cells, as well as the purity, safety, functionality, and genetic stability of cultures grown from those banks. From a genetic QC perspective, it will be important to look into the genomic coordinates of integration site or sites, transgene conformation and flanking regions in gene of interest, as well as into expression, construct integrity, and vector copy number also must be reported. In fact, these characteristics are also requested by regulatory bodies and highlighted in ICH guidelines. At Cergentis, we offer an extended report which was prepared together with a regulatory consultant, and it also meets regulatory requirements and can be used directly for the filings. In this overview table, we summarize the key characteristics that need to be looked into as recommended by guidelines from the FDA and EMA. So basically, these regulatory agencies, they want to ensure that all biologics are of the highest quality, Therefore, testing specific critical quality attributes that have an important influence on the consistency and safety of a product is mandatory. As just discussed in the previous slide, genetic characterization needs to be performed not only to mitigate the risk of downstream processes, but also because it is requested by regulatory authorities. So the most relevant genetic parameters you can see summarized here on the left side of the slide. And on top of the slide, you can see a range of different technologies which are used for the characterizations. And of course, all of these technologies, they have their advantages and limitations, and it really depends on the research questions you want to answer. But basically, each method allows insight into only one or few parameters of interest. But to get a comprehensive picture, you will need to use a combination of the several methods. So a key distinction of our TLA-based approach is our ability to check both transgene integration sites and the integrity of the integrated sequence in a single experiment. However, as you might know, TLA only provides an estimation for the copy number, and since the FDA puts a strong recommendation to determine the precise vector copy number, we have worked on the solution to determine it. So by coupling TLA-based data with ddPCR results, we are able to get the precise vector copy number as well. If we move forward and we talk about the clonality, so both industry and regulators want to ensure consistency and quality of biological therapeutics, and therefore their manufacturing process. And according to officials, one critical criteria here is to assure clonality. So clonality assurance is needed to minimize the heterogeneity of cell banks and therefore favor a consistent process to manufacture of a product. And in fact, the FDA and EMA in their guidelines both explicitly indicated that biologic products should be derived from a single cell progenitor or monoclonal cell line. And however, while recent developments like single cell sorting and high throughput imaging allow for the acquisition of evidence supporting clonality during, for instance, an IND or BLA filing of newly generated cell lines, such acceptable assurance may not be available for the legacy cell lines. In those cases, recloning of the MCB late in the development may introduce significant risk, such as affect titer and quality of the products, and might also not be advisable due to comparability issues. So here what you can see in table 1, this table outlines some alternatives for genetic testing that can be applied toward clonality assessment of a legacy cell lines. However, besides a lower resolution, none of these listed technologies can output all the essential genetic characteristics in a single experiment, like TLA-based assays can. In fact, we have, in collaboration with Novartis, developed a high-level statistical analysis to better assess clonality in recombinant cell lines. And the outcome of this collaboration was put forward in a joint white paper. So here I would like to look into a bit better how our clonality package looks like. So our clonality package consists of 2 parts. In the first one, we perform the comprehensive genetic characterization of a cell bank, for example, master cell bank, using our TLA technology. And during this part, unique integration sites of a master cell bank are detected. In the second part, we use genetic data from the first part to design specific primer sets to detect 2 of a master cell bank specific breakpoint sequences to be able to screen number of subclones deriving from master cell bank in the qPCR assay. So basically, if a cell line is clonal, we expect that all subclones will be positive for the tested qPCR assay. And with this type of approach, we will give a probability of 96.8% that the cell line is clonal with 95% of a confidence. So as mentioned in the previous slide, proof of genetic stability is also requested by regulatory authorities. So it is important to show that all of genetic characteristics are stable across culture generations and that genetic drifts do not compromise the consistency of a product. So here we would like to share with you examples of comparisons between master cell bank, working cell bank, and end of product cells of different cell lines. So as you can see, the working cell banks and end of product cells of cell lines 1 and 2 share all integration sites, single nucleotide variations, and transgene fusions identified in the original master cell bank. So this data suggests that cell line 1 and 2 are stable. Their genetic differences occur in cell line 3, so in which 2 integration sites, 2 single nucleotide variations have arisen, and 2 transgene fusions are lost over time. So with this slide, I would like to summarize what we have discussed. Basically, genetic QC is essential in analytics and product characterization. Genetic QC is requested by regulatory authorities. It helps de-risk R&D program decisions, as well as supports consistency and quality of biopharmaceuticals and patient safety. And with our TLA-based solutions, we can support you in every step of the cell line development process. Since this year at Cergentis, we offer several different solutions based on which stage of a cell line development process you are. And every Office package comes with its advantages. So for example, if you are at your clone selection stage, we can help you to identify the sibling clones and select the ones that contains no undesired mutations. If you would like to speed up your clone selection process, we also have in-house solution for the CHO cell clone selection. And the main benefit of the 3 packages listed below is that we come with an extended reporting, which can be used directly for the filings. And also cell bank characterization, as well as genetic stability package. They are also combined with ddPCR analysis to precisely determine vector copy numbers. And besides of the predefined solution packages, we are also happy to discuss any projects which are out of our standard offerings. So our custom assay development team is happy to discuss any challenging projects you might working on. And as just mentioned, since this year we have implemented ddPCR assay, which can be performed as a standalone assay, also together with a TLA. And if you would like to learn more about ddPCR, please check out our webinar, which is available on our website. So with this slide, I would like to point out that our services cover the entire life cycle of drug development process. Since last year, Cergentis has joined the Solvias group. Solvias has a comprehensive analytical testing portfolio and capabilities to support all stages of drug discovery, development, and commercial release. And the acquisition of Cergentis strengthens Solvia's platform of biologics and cell and gene therapy testing solutions. Here is an overview slide touching upon our main expertise, in-house capabilities, and experience at Cergentis. So at Cergentis, we have a dedicated team of PhD level scientists. We are also ISO accredited, which demonstrates that we operate our services within a robustly reliable compliant framework. We have a pleasure to support leading biotech and pharma globally in over 40 different countries. And with over 10 years of experience, we had the pleasure to make world-class partnerships And also our unique genetic insights continue to heavily endorsed in peer-reviewed scientific publications. All of those are available on our website if you would like to check. And during this presentation, we have talked about several gene editing methods, but in this slide, I would like to highlight that at Cergentis, we can adapt to your needs. So we have a lot of experience with different gene editing tools, such as transposon-mediated integrations, CRISPR, as well as random integrations events. Also, during this webinar, we have mainly discussed CHO cells, but we have experience working with different cell lines. Also, something completely different from what we discussed today, we also work with animal models as well. And at Cergentis, we offer end-to-end service. So basically, we only request from you to prepare the samples, and we will take care of the rest, so including shipment of the samples, primer design, lab work, NGS, data analysis, as well as report preparation, which is shared by one of our experts. So with this slide, I would like to thank you all for your time. We hope that this webinar has been useful and informative and has given you a better understanding of genetic characterization needs during cell line development process. With that, I would like to conclude and thank you all again for joining us today.